Paul actually asked me to talk about something fairly simple that I've yammered on about over the years a fair amount, and that is the opportunities for synergy between the DNA barcoding movement and long-term ecological research sites. Many of us are collecting in, sampling in national parks or protected areas or whatever, and in this particular case, I'm talking about a specific subset of those which host long-term ecological research programs and infrastructure. It might be of a more sort of biodiversity sense. It might be more of a physical environment sense, like these sites that host hydroflux towers and things like that. There is a formal network of so-called LTER sites both in a number of countries as well as internationally. In the United States, we have something called the National Ecological Observatory Network, which is a network of some 60 sites that are very heavy on physical science instrumentation and kind of light on biodiversity science, but we're working on that. And the example that I want to talk about here today a little bit is a network called ForestGEO. It used to be called the Center for Tropical Forest Science, but as temperate zone sites were added, they had to change the name. It's a network of, right now, 67 large plots. They're usually 50 hectares in size in 27 countries. Their core activity is studying 6 million trees, which is about 13,000 species, which is something like a quarter of the world's tree species, depending on how you count. They are sort of all over the map. We actually heard yesterday about a desire to do this kind of work in the Palawan site in the Philippines. There are some significant gaps in Australia, parts of Africa, parts of Latin America that there are efforts to fill. The core methodology at these is basically any tree or stem that's sort of more than a centimeter in diameter gets inventoried, identified, mapped, measured every 1 to 5 years, and that provides a tremendous amount of information that you can kind of think of as a set of GIS layers, that then you have soil, microclimate, various other things. So it provides a very information-rich context in which to do other things, even if you don't care about trees. But there has been some DNA barcoding effort in this network. John Kress and others have done complete DNA barcoding of the plants in 15 plots and made that available. Yves Basset out of Smithsonian Tropical Research Institute in Panama is running a sort of pilot program for arthropod monitoring and has done some barcoding as the backbone for that in Panama, Thailand, and Papua New Guinea. And I want to talk about some very preliminary data that we have from malaise traps. And I will say that some of this data was just posted on iBOL last week, and there's a lot of sequencing that is continuing, so therefore there's no real analysis of this yet, but we have run traps in global malaise trap protocol traps in one temperate zone forest in Virginia, in the United States, one tropical savanna site in Kenya, and 3 tropical forest sites in Papua New Guinea, Thailand, and Panama. These were all run for a year. One of them is still running. In most cases, we've analyzed every other week of sample. And that, of course, is one of the questions that needs to be looked at is what's the sort of trade-off between information content and sampling intensity. When I think about barcoding, one of the things that's really exciting to me is the synergy between field sampling and research and monitoring activities on the one hand, and museum-based library building on the other, so you can really merge that sort of historic information with what are the critters doing today. So if we do that in the Front Royal Virginia trap, which is about 70 miles west of Washington, D.C. in the Appalachian Mountains, we have, as I mentioned yesterday, been intensively building a barcode library for Lepidoptera, butterflies and moths for North America. On the other hand, that has not happened for Coleoptera, the beetles. So what's the influence of that when you put out a Malaise trap? So for the Lepidoptera, 14% of the bins have no species name. Now if you look at that, those are mostly really tiny, nasty microlepidoptera like Galethioid moths, and there's actually a lot of specimens just behind us that are in the process of getting sequenced that represent those names. So I think that we're going to reduce this number. 65% of the bins have one species name available in bold, which is a good thing. Interestingly, 24% of the bins have 2 or more species names in bold, and that's, I think, coming from 2 different things. One of them is a case where there's a misidentification. So you have 60 sequences that have the right name and a couple of them that have a wrong name, and that's just a curation and cleaning problem. And then there also are some areas where there's a species complex and there is work that has to be done to resolve which is the right name. So if you move down to beetles, where there has not been a systematic library building effort, at least at the level of North America overall, 50% of the bins have no species name available in bold today. 45% of them do have a species name, one species name available in bold, so we're actually doing pretty well. And 5% have 2 or more species names available. An interesting application of barcoding in this that actually Rodolphe was involved in this. One of the target taxa that Yves Basset has been working on monitoring in some of these tree plots is the Saturniidae, the wild silk moth. He's done that for 6 or 7 years doing light traps, been using DNA barcoding as the taxonomic backbone for that in association with Rodolphe. But in this particular analysis, in addition to that recent light trap data, we were able to go into museum collections and look at earlier records of additional species that were known from the site historically and look at change over time, and go into historic voucher specimens from Annette Aiello and others' life history studies, and in fact some from ACG, and look at, what do we know about the life history of these things. So the taxonomic, DNA barcode was used as the taxonomic backbone that brought all that together. It has also empowered studies that Eve and others have done of ecological interactions rearing insects from fruits and such things, which brings me to my conclusions, which basically are, I think that these so-called LTER sites provide an opportunity as we think about how to roll out sampling programs because of the synergies with other kinds of activities. They show the value of the intersection of field sampling and museum library building activities. And if we build these kinds of collaborative knowledge bases, then of course also we're laying the foundation to do eDNA and metabarcoding and use kind of the next generation of tools. And just in closing, thanks to the folks here in Guelph and the ForestGEO people and lots of other collaborators who've helped develop those ideas. Thank you.